So hi everyone, welcome back to our lectures going over neurodevelopment and neurodevelopmental disorders. And today is part 2 of the Posterior Fossa Malformations lecture, where we will cover Joubert and syndrome and cerebellar heterotopias and dysplasias. Again, these are meant to be educational lectures for students and residents, where we focus more on the conceptual aspects of the disease, and these are not meant for diagnostic purposes. If you are suspicious or one of your family members or you may have one of these disorders, it's best to talk about it with your doctors on how to best manage it. In the previous lecture, in part A, we talked about Chiari and Dandy Walker malformations, and now we're going to wrap it up by going over Joubert syndrome and cerebellar heterotopias and dysplasias. And again, the key really is that some of these have different genetic and molecular bases, some that recur in families, some that don't. So really accurate classification is really meant to help in family planning and to hopefully help us better understand the pathophysiology of these rare disorders so we can potentially develop therapies one day. These are the books that were used to generate this presentation. These are the books that I used during my training, especially the one on the left, just because it's a little bit older. So please do supplement your readings with these great books. So one of the things that's important to understand both Joubert and some of the cerebellar dysplasias is really the concepts that surround cerebellar development embryologically. So around the first month of life, just as that neural tube is forming, you already have some hints of cerebellar starting to develop around the just below the mesencephalon and the kind of metencephalon area, where you have what are called these cerebellar primordia. These things here that are kind of lateral to the neural tube, around that kind of hindbrain area. And these are really going to form the cerebellum under the influence and signals of this isthmus organizer. And it's important to understand these concepts because we'll see them emerge in many disorders that we talk about today. So basically, there is this structure, just like the nodal cord in the that instructs the development of the neural tube, that secretes some factors. It namely secretes FGF and Wnt, and does so in this kind of metencephalon, rhombencephalon area in the back of the brain, around the junction between those two structures. And it's going to secrete a number of factors that help guide patterning. And really what these do is that they instruct two separate types of cells. We'll see these cells in different frames of view. So if this doesn't make sense, don't worry, there's another image. They'll instruct the ventricular zone to form GABAergic neurons through radial migration. The chief neuron we're going to be talking about that's GABAergic in the cerebellum is the Purkinje cell. And then dorsal in the back, lateral on the outside, the differences in gradients will lead to other precursors adopting different identities. And these are going to be the cerebellar nuclei neurons in the pons, inferior olivary nucleus, pontine nuclei, and then a separate population is going to form the granular cells, the glutaminergic granular cells of the cerebellum. These become the external granular cell layer through tangential migration, and then they migrate internally through the Purkinje cell layer to become the granular cell layer of the mature cerebellum. And what I find most interesting is that in other parts of the brain, the GABAergic neurons are the ones that will tangentially migrate, and it's the glutaminergic neurons that will radially migrate. Here, it's almost the opposite. So just keep that in mind. Why that's the case, I'm not sure. Could just be structural, but needed to say, I thought that was quite counterintuitive. So, let's see what the notes say about these concepts. So at 9 weeks, you have two proliferative zones. You have the dorsal medial ventricular zone. These are going to, as we said, migrate radially to form the GABAergic neurons of the roof nuclei and the interneurons, and then the Purkinje cells as well. And then you have the rhombic lip, which is the dorsolateral subventricular zone, and these are going to produce the glutaminergic neurons that tangentially and inwardly migrate to form the cerebellum, the pontine nuclei, and olivary nucleus. So here is another structure that just shows this. This is an axial section through this through the pons and developing cerebellum around 14 weeks gestation, and you have this blue ventricular zone here where radial migration is going to lead to the Purkinje cells and the interneurons of the dentate being formed, and then you have this rhombic lip that's divided by this choroid plexus. The posterior part's going to tangentially migrate to form the external granular cell layer and also contribute some glutaminergic signaling to the dentate. And then the other the other part of the rhombic lip will migrate inwards to form the olivary nucleus, the pontine nuclei of the pons. So this is another photo showing the kind of evolution of the external granular cell layer. So it's about 20 to 30 weeks gestation where you start seeing foliations macroscopically. And really, this is the peak thickness and proliferation of the external granular cell. It's about 6 to 9 cells thick. And really, it's just after the Purkinje cells start to become discernible. So here they are. Interestingly, they release sonic hedgehog, which is thought to cause the proliferation of the external granular cell layer. So these coincidences they coincide in visibility because as the Purkinje cells mature, they will stimulate the external granular cell to proliferate, and then once that's done, they will start migrating inward and fill in this internal granular cell layer. So there's a number of synapses that start to form as well. And of course, there are theories of that synapses will create some tension to create foliation. So that's also something to consider of kind of these coincident timings of many events. But the synapses of the Purkinje cells will form with the internal granular cells that will form synapses with the Purkinje cells through these fibers called parallel fibers. These are driven by what are called Berman glia, which are the radial glial cells of the cerebellum. And again, these have an important role in many neurodevelopmental disorders when they become defective. And we'll see that in some of these disorders. Mossy fibers, these are fibers from the pons that connect the pons to the pons to the internal granular cell layer through the middle cerebellar peduncle. You have climbing fibers from the inferior olivary nucleus and the spinocerebellar tracts that connect to the cerebellum through the inferior cerebellar peduncles, and then the roof nuclei will exit the the cerebellum through the superior cerebellar peduncle. So some good anatomy review here, but it's important to note that all of these are occurring in the kind of end of term 2 years of life. Particularly for the vermis, this is one of the last things to develop. And again, this is facilitated, the foliation is facilitated by Bergman glia that really sit in that Purkinje cell layer with processes extending into the molecular layer and the internal granular cell layer. So you can imagine there's going to be lots of things radially migrating along these fibers. These arise in the ventricular zone through the kind of definitive radial glia, and again, aid in the migration of the external granular cells and Purkinje cells through migration. Now, they rely on a number of signals to be to develop properly and to develop in the proper number. Sonic hedgehog, FGF Wnt, and so if there is a defect, they're going to not be able to do these important steps of connecting neurons to one another to improve foliation. There's not going to be enough to facilitate the growth of the cerebellum, and usually the structure affected is the vermis in these cases because it is the last part of the cerebellum to develop. So you can imagine that if there is an exhaustion of these internal granular cells and Purkinje cells, the final structure is going to be affected the most. Also, their fibers, their highways for migration. So if there is some anatomical disruption in their structure, you're going to have dysplasias and migratory defects in the cerebellum. And the reason why, again, that these are so evident is that the cerebellum dramatically increases during development. It increases about 2 to 5 times in weight and about 30 times in surface area in that final stage of development. So perhaps these cerebellar things, you can imagine that you can have a fairly minor molecular defect, but because it's so late in development and requires so many steps, you can have a relatively normal brain and have some defects in this structure. So with that in mind, let's see how we can integrate that neurodevelopmental kind of playbook into some of these disorders. So we'll start off with Joubert syndrome. This is a genetic ciliopathy. So we're going into the mechanism right from the get-go. Cilia, as we'll see in a few slides, really help coordinate how wind and sonic hedgehog interact with, communicate with one another. And so if you have defects in cilia and primary cilia, you're going to disrupt the normal communication between cells in this critical period. Patients have hypotonia, ataxia, abnormal eye movements, abnormal breathing patterns, and intellectual disabilities, which can overlap with other disorders, but these are the kind of classic symptoms of Joubert syndrome. And then on imaging, they have this molar tooth sign, where you have this kind of molar tooth-like structure between the cerebellar peduncles and the lack of a vermis that is thought to be kind of pathognomonic for this disorder. There's a number of non-neural features as well, but the brain malformations include vermis, hypoplasia, abnormal corticospinal tracts with them not crossing, and there's a number of genes that have been implicated, and most of them seem to coincide with cilia function. Now, There is something called Joubert syndrome related disorders. These are disorders that have the molar tooth sign, but do not have the classic clinical symptoms. So just something to keep in mind. And again, the key is, as we'll see, they may have different genetics and have different implications for families. So this is why it's important to sort out exactly if it's Joubert syndrome or not. Very rare disorder, 1 in 100,000. So this molar tooth sign, it's visible 17 to 18 weeks. Around that time when cells are forming these different layers in the cerebellum, things are connecting through these peduncles. Very visible early on. And really the best way to see it is through these axial sections of the upper pons and ventral midbrain. There's cerebellar vermis agenesis. The vermis, again, is a very sensitive structure because of its late appearance. And there could be rotated and think superior cerebellar peduncles. Again, just due to disruption in the normal developmental process. The intrapeduncular fossa is widened, is deep, and there could be widening of the cortical spinal tracts because they're not crossing. Ventriculomegaly, retrocerebellar fluid brings the possibility that this could mimic Dandy Walker malformations. Sometimes you can have both, but again, this is a autosomal recessive disorder, and some of the other disorders that may mimic are sporadic or have different genetic contributions, so it's important to separate them for family planning. So here are some pictures. We have this enlarged cerebellar hemisphere, in this case here, wrapping around the spinal cord, and then you have this cyst-like structure here that may mimic Dandy Walker malformation. When you section it, though, you have this kind of molar tooth sign here, vermeil atrophy, and then this kind of bat wing-like triangular shaped, umbrella shaped, whatever you want to call it, fourth ventricle. That's quite characteristic. So agenesis of the corpus of the cerebellar vermis. Hemispheres can be normal to atrophic or even larger, as we see in this picture. And there's white matter heterotopias, including in the Purkinje granular and deep nuclei neurons. And this may kind of help us understand the pathophysiology, that this is potentially a migration disorder of disruption of this isthmus and organizer and the ability of the cells to communicate through these important signaling pathways. Fragmentation of the dentate into smaller islands, and the brainstem again could be have these crossing cortical spinal tracts, widened intrapeduncular fossa, and then multiple abnormalities. Most of these I think we've covered. Fragmentation of nuclei, hypoplasia of pons and olivary nuclei, and disorganization of the posterior medullary structures as well. So here's a picture of the cortical spinal tracts forming these very parallel fibers, and here they should be desiccating, and so very interesting image, to say the least. Here we have these fragmentations of the dentate nuclei that we just talked about, and then you have what are called posterior medullary protuberances, where you have the various abnormalities of the medulla. Here we should have a we should have a sulcus separating the left and right posterior medullary areas through a sulcus. this is missing, and then we do not have we have disorganized the kind of dorsal region nuclei, the nuclei gracilis and nuclei cuneatus is not easily seen. And again, the crossing of the fibers, these are going straight down. We don't get a sense that there's any crossing at all. So lots of abnormalities in the brainstem as well. So here is that cilium that seems to be the key the key driver of these disorders. So these are typically autosomal recessive. So it means that if you have a child with Joubert syndrome, there's a 1 in 4 chance that the parents can lead to a similar offspring through this autosomal recessive fashion. There's about 30 genes implicated in this disorder, but there are some rare de novo sporadic cases, and so they don't all follow the autosomal recessive, but that's the typical pattern. 62% of cases have causative genes identifiable through exome sequencing. There's not one gene responsible for more than 10% of cases, but they all have this unifying thing of being involved in cilium biology. So primary cilia, these are non-motile cilia. They have this 9+0 structure where you have these 9 rods that create some sort of structural support, but they lack the +2 dynein arms that are involved in motility in the motile cilia. And again, these are usually involved in preserving some polarity and cell signaling function. And so when they're lost, you can imagine that a lot of those neurodevelopmental things that we discussed are going to be perturbed. And when these happen, we're going to get alterations in many of these structures. And that's why all those structures are affected, because the signaling from the isthmus organizer and the migrational patterns all rely on those signals. Okay, so let's move on to cerebellar heterotopias and dysplasias. This is what we're talking about, these kind of ectopic gray matter structures, usually found in the white matter. And these are really a spectrum of neurodevelopmental disorders, from anywhere from severe to asymptomatic. So quite a large spectrum, and the extreme cases are extremely rare and usually occur with other CNS and non-CNS organ malformations. Usually the cortical stuff is more severe, and so these become a feature of another disorder focused on cortical abnormalities, and so it's very rare to make a cerebellar diagnosis of heterotopias and dysplasias in isolation. There's 3 main categories. There's cerebellar heterotopias of infancy, where you have displaced neural and glial cells, often in the white matter, as we see here. These can be asymptomatic, so you may see them in otherwise normal individuals. You have kind of a second category, which are known as dysplasias associated with more extensive cerebellar malformations. And these are usually where the cerebellar heterotopia accompanies another major disorder, and it's just a feature. And then we'll talk about one of the major dysplasias, the Rhombencephalosynapsis, where you have vermeil agenesis and an apparent fusion of the cerebellar hemispheres, with very classic findings. So let's start with the least symptomatic one first. Cerebellar heterotopias of infancy. So here again, you'll see these misplaced nodules of cells, usually in the white matter. Interestingly, they stain for the same markers you'd get in their normal location. So you have synaptophysin, where these little granule cell-like neurons may be forming synapses. And then interestingly, you have GFAP around them that suggests that these are not just disorganized, some of these at least, are not disorganized random collections of neurons, but they're forming functional connections And the significance of the glial cells may suggest that abnormalities in glial cells may have led these granular cells to migrate to the wrong location. So offering some potential insight into the pathophysiology as well. So by definition, they're misplaced and disorganized collections of neural and glial cells, and they're usually associated with other migrational disorders, which may suggest that they're in fact they're true migrational disorders where neurons migrate along ectopic astrocytes or vessels. So any problems with radioglia, any problems with an insult that leads to specific radial fibers of Bergman glia misaligning, you can get these structures. And animal models suggest that there's possible disruptions in these kind of developmental pathways that may result in kind of premature differentiation of granular cells. If they mature before they reach their final location, you may get these heterotopias. So these are very, as the name suggests, they're very common in infancy. They can be found in otherwise 50% of normal infants. But usually because the cerebellum expands so much, these are going to get drowned out in adulthood and become less frequent. So they're asymptomatic, and so if you see them in isolation, do not be alarmed. Do not obviously, it's important to think about other disorders, but them in their own is not enough for a specific diagnosis. If these persist, there is some kind of interest in understanding if these are related to neoplastic processes, but we'll leave it at that. And then they are more common in certain developmental disorders, so they do have some genetic basis. So there's multiple ways to describe these patterns. You can get these compact groups of just mature neurons. These are usually in the dentate or the adjacent white matter. You can have immature collections of granular cells, usually, as we saw, perivascularly or focally because of abnormal migration down the wrong kind of highway. And these, again, just may represent mismigrated cells from granular cells on their way to the internal granular cell layer. And then there's some more formal definitions called heterotaxia of Brunn. These are usually terotaxis of Brunn. These are poorly disorganized mixtures of cell rests of immature and mature neuroepithelial cells. And then you have heterotopias of Brunn, and these are really mixed cell rests with normal relationships of the cerebellar cortex. So if they're disorganized, they're heterotaxis. If they're organized into some more advanced structure, they're called heterotopias of Brunn. Here's an example here where you have these granular cells around a blood vessel. You have a molecular layer and then a granular cell layer, or vice versa. That seems more organized, so that would be an example of a heterotopia. Here we have a kind of a focal disorganization of the cerebellar tissue. We have these kind of perivascular collections of these granular cells. We have these kind of collections of immature and mature cells here. We talked about this one here. This was called the heterotaxis in the book. You have mixed cell rests. Some of these look much more immature than the normal structure here. So perhaps that is the reason why that got labeled that way. And then you have this kind of fragmented dentate there. So the second group is really cerebellar dysplasias associated with syndromes. This is the table that kind of shows which syndromes are known to have cerebellar dysplasias that are quite common. We'll go through them, but again, Usually the cerebellar dysplasia is more severe, but it's usually a feature of a larger syndrome. And as we talked about, the cortical manifestations are just so severe that the cerebellar aspects become more of a feature than the actual defining feature of that disorder. They can involve the vermis, the hemispheres, or the entire cerebellum. And as we'll go through them, you have a number of lissencephalies, as we'll see, that have cerebellar findings, especially the ones involving renin, as their name suggests, lissencephaly with cerebellar hypoplasia. You have syndromes with occipital encephalocele like Meckel-Gruber. These may be more structural than actual neurodevelopmental disorders of migration. And then you have numerous other kind of disorders, metabolic, Zellweger syndrome, isolated hypoplasias of the vermis, Dandy-Walker malformations that may also have some of these severe dysplasias. Again, some of these could be mechanical, some of them could be part of the pathophysiology. And then you have Rhombencephalosynapsis. So just to rehash some of those, so there is pronounced cerebellar dysplasias in cerebro-ocular dysplasias, especially those associated with type 2 lissencephalies. These, as we know in other lectures, are defects in glycosylation. A Walker-Warburg syndrome is one of them. And basically, cerebellar dysplasias is part of the criteria of some of these disorders. So do look for them if you are suspicious of these disorders. And we see some of those similar aspects that we just talked about with white matter heterotopias, simplification of that dentate and vermial hypoplasias. Cerebrocular muscular syndrome, again, part of that kind of type 2 lissencephaly. Fukuyama congenital muscular dystrophy, which are these type 2 lissencephaly associated with these glycosylation defects. And then there is a type 1 lissencephaly with striking cerebellar hypoplasia that's due to mutations in renin. Occipital encephaloceles like Meckel-Gruber, this is likely thought to be more of a disruption than a true malformation. And then Zellweger syndromes, defects in peroxisome metabolism, and these are associated with extensive migrational abnormalities. Finally, we're going to talk about rhomboencephalosynapsis. This is a very rare malformation characterized by vermial hypoplasia and apparent fusion of the cerebellar hemispheres to form this pear-shaped cerebellum. This is not associated with a fourth ventricular midline defect to help it differentiate it from some of the other disorders we talked about. May include fusion of the thalami, inferior colliculi, and then usually associated with other cerebral defects and death early in life. Gross, you have this small pear-shaped cerebellum. You have aplasia of the vermis with an apparent fusion of the cerebellum's hemispheres. This diamond-shaped ventricle that may lead to hydrocephalus. And I think that's why it's important to look for that fourth ventricle defect so you don't mistake it with other disorders that have hydrocephalus. But it has a very distinctive microscopy when it's a classic case. So it's a spot diagnosis. Has this U-shaped dentate nuclei surrounding the fourth ventricle. And again, you can have other defects in these structures that are formed from this isthmus organizer. So here is that kind of classic U-shaped dentate that doesn't separate into two different nuclei, but just kind of surrounds the fourth ventricle. This is very easily a question you may get on a board exam. So what's the pathophysiology? Again, it's a defect in this isthmus organizer. There's different gradients in neurodevelopment. Some come caudally, some come rostrally. And when there's an intersection of these signaling molecules, this will set up the isthmus organizer that is responsible for secreting that FGF and Wnt are responsible for proper cerebellar development. So again, this structure is formed by the rostral and the caudal and rostral limits of these homeobox genes OTX2 and GX2, which help form this isthmus organizer. It will then secrete the right molecules to help the cerebellar form. And if you mess with these dorsalization factors in animal models, you'll get something similar to Rhombencephalosynapsis. And the severity will depend on how profound this defect is. So, the tying aspect of all these things is a defect in the isthmus organizer, a defect in its ability to communicate with the cells through these primary cilia. So hopefully, as we better understand the biology, we'll be able to devise some meaningful therapies for these patients. So thank you very much. That concludes the second part of our posterior fossa malformation lectures. I hope you found that useful. I hope you learned some nice embryology and understand how defects can lead to very severe neuropathological disorders. And hopefully that provides you a framework to remember some of these for your exams and for your careers, of course, because these are very rare. You may only see these once in your lifetime, and it may be, if you can recognize it, it may make an important impact for patients and their families.